Okay. So when we left off, we were talking about equilibrium potentials and how they're related to the electrochemical driving force. Let's do a little practice problem with that first. So I want you to go ahead and draw this situation, do anything that'll help you to answer this question. We're thinking about sodium with an equilibrium potential of positive 60. We're going to try and use that information to figure out what direction our electrochemical driving force, our ECDF, is going to be if we start with a cell that's at a membrane potential of negative 70. We're going to share a tool you guys can use to practice this in the future in a second, but let's walk through this example. Okay. So I made myself a little picture of our setup. So you might draw something like this anytime we're thinking about an example that involves our driving forces, our gradients, right? So this has everything that you could possibly have in a question that you might be interested in, right? So we're thinking about some specific ion. We might, in this case we don't, but we might have information about concentration in extracellular fluid and intracellular fluid. We might, in this case I drew on our membrane potential, we might have information about our membrane potential, in this case negative 70. We might have information about our equilibrium potential for that ion, and we're usually going to be asked to solve for one or more of our driving forces, whether it be electrochemical driving force, we have our negative bottom there, or our electrical driving force, or our chemical driving force. Okay. So in this example, we're working with sodium. Okay. So I'm going to go ahead and fill that in. All right, so we're working with sodium, a cation ion. We have on here that we're thinking about a cell at rest, meaning that we have a separation of charges across the membrane such that we have a membrane potential of negative 70 because there's that many more negatively charged ions, so that many more anions inside our cell versus outside our cell. Okay. So in this example, we also know that we have an equilibrium potential for sodium of positive 60. So we defined our equilibrium potential last week. Our equilibrium potential is the membrane potential at which our chemical driving force and our electrical driving force are going to be the same size, so the same magnitude, so equal, but they're going to be in opposite directions, leading to a total electrochemical driving force of 0. So when this cell gets to positive 60, there's going to be no force overall, so no electrochemical driving force on sodium. But if we're not at positive 60, there is some force acting on sodium. And that force is going to go the direction that leads us towards a cell with a membrane potential of positive 60. So if we're starting at negative 70, we want to get to positive 60. We know that sodium is a positive ion, cation ion. our electrochemical driving force is going to point. Okay. So what we're imagining here, that if we're starting at -70, sodium wants that membrane potential to get all the way up to +60. You're going to have to move a lot of sodium in to do that, right? So every time you let sodium in, that membrane potential gets more and more positive, right? So closer and closer to zero. And in fact, this electrochemical driving force is so strong that we get above 60. We get all the way up, sorry, we get above zero. We get all the way to +60. So this is a really strong force here. I picked this example because this is going to be super important for neurons in our next unit. This is going somewhere. So that's our total force, right? So our electrochemical driving force is our total force on the ion. So we start out with a situation where there's some sodium inside, some sodium outside, and on net, that sodium is going to want to move into the cell rapidly a lot until we get to +60. But that electrochemical driving force is composed of two other forces, right? So we could break it down into two separate pieces that add together to make that big total force. And we can think about what those are in this example as well, although I didn't ask you. Okay. So in this case, looking at the charges on the sodium, so a sodium positive cation, our membrane potential, that negative polarity, we're at -70. That makes it super easy to recognize our starting electrical driving force. Because with our electrical driving forces, we've got opposites attracting, right? So opposite charges attract. So our electrochemical driving force points in. Our electrical driving force points in just like our electrochemical driving force does, right? So that positive is attracted to this negative. Okay. From what I've given you, figuring out the chemical driving force is a little trickier. We could do it, and we'll think about this more as we think about the setup of neurons in the future. But I'll just mention it right now to be complete. All right, so one of the hints for this, you can see from the size I drew these arrows, right? Our electrochemical driving force is really big, really long arrow here. Drew the electrical force, that's slightly shorter, slightly smaller in magnitude. And that's because the chemical driving force also points in, right? The electrochemical driving force, that total sum force, is so big because we're starting out with two forces that are both going the same direction. So they add on top of each other. It's like a multiplier. If you didn't have my arrows on here, another way you could think about that, if you want, and if this confuses you right now, forget I'm about to say what I'm about to say. But if you think about the situation when our membrane potential gets to say +30, right? We're not all the way to +60. Right, at that point, our electrical driving force would be in the opposite direction. It would flip-flop when the polarity of the cell changes. But we know that that electrochemical driving force is going to be pointing in until we get all the way to +60, which means we need something other than the electrical driving force to explain that. And in this case, it's a really big chemical driving force. It's a huge difference in concentration gradient. So we set up a situation using sodium potassium pumps where we have a really high concentration of sodium outside. Actually, I've drawn it in the little bucket. Right, so high concentration of sodium outside and a lower concentration of sodium inside. So at this point, I wouldn't expect you to step forward and imagine that from this example, but if I give you that high concentration or any numbers related to that, at that point, you should be able to tell the direction of the chemical driving force. Okay. So the other thing that I'll post in our course after class is I have a link down here for myself. This tool that I created, I made a little spreadsheet with some little calculations, some little functions in those boxes so that you can practice. So I know we don't have that much time to go through tons and tons of electrochemical driving force practice questions in here. But it's the kind of thing where once you get it to click it clicks. But in order to get it to click, you might need to go through lots and lots of examples. So I've made something where you can type in an existing membrane potential of a cell, right? So in this case, -70, right? So like our sodium example right here, you might put in then the equilibrium potential of the ion. In this case, we know that's +60. Right? And then you'll also have to fill in the final thing we need to think about, which is the charge on the ion. Positive here, right? And it'll fill in and tell you what direction the electrochemical driving force is for that ion. So you can use this to practice, make up any numbers that you want, should get you the correct electrochemical driving force for that membrane potential, equilibrium potential, and charge on the ion, whether it's an cation or an anion. And I recommend just doing it until you are consistently predicting the correct direction for the electrochemical driving force. I'll give you guys a copy. Okay. So we can set up lots of questions with that. We do also know some things in general about what concentrations of solutes are going to be in typical intracellular fluid and extracellular fluid here. So you can see here, if you consulted a table like that, you would know about that sodium concentration. Right, so here we see that huge difference between extracellular fluid concentration of sodium and intracellular concentration of sodium. I don't expect you to memorize this table. So if you need that information, I'd give it to you. But these are things that we can play with as we're coming up with practice problems and ways to think about this. So here's some questions for you from your book related to this topic. We're going to move on to our next piece. We're going to be thinking about the movement and the rate at which our substances, like ions, might be moving across membranes. Some things that aren't ions. That's why we've been setting up this idea of the electrochemical driving force, because we need to know, in order to determine whether something is passively or actively transported, which direction it wants to go in first, right? If it wants to go in that direction, it's easy, you're not going to need energy. If it doesn't want to go in that direction, if there's a force pushing it in the opposite direction, you're going to need to get an energy source from somewhere to overcome that. So we're going to start by finding transport. So when we're thinking about transport, we're thinking about net movement of our molecules, of our substances. Right? So we're thinking about what we might call flux, as we see labeled in this image. Right? So we're thinking about directional flux. So in this top image, we can see flux in one direction, right, going one way. In the bottom, we can see flux going the other way. And this top example, this top figure, is showing us a system that's at equilibrium, right? So everything kind of evenly spread out. If you look at those concentrations, you can see there's no concentration gradient, there's no difference, right? So here we have 1 molar blue dot on side 1 of this example. We have 1 molar blue dot molecule on side 2. They might be able to move across the membrane in the middle. In this case, they can. But in total, anything that moves across one way is going to have something randomly balancing it in the other way, because we've got this equilibrium state, we've got this even concentration on both sides. So this is diffusional equilibrium, meaning that there's no net, so no total transfer of molecules. At the bottom, we see something where we do have net flux, so net movement of our molecule or our particle here. Right, so you can see that there's a lot more movement from side 1 to side 2 than there is from side 2 to side 1, giving us a net flux, right, so a net transfer of particles from side 1 to side 2. And if we map that onto our understanding of concentration gradients, we can see why. Right, so here is that high-low concentration that we're looking for with CDF, our chemical driving forces. Right, so here we see our high concentration, 2 molar. Here we see that low concentration, 1 molar, right, giving us that total net direction. Right, this is the setup we're kind of doing shorthand for when we do that. So pulling this and combining it with our electrochemical driving forces. Let's think about electrochemical driving forces for sodium, which we just did, and try and link this up to the word active and passive. You've seen in your prerequisite courses, I hope, but we're certainly about to talk about. So if we've got an ECDF for sodium pointing into the cell, is our net flux of sodium from the ECF into the ICF going to be active or passive? Okay, so what we're describing in this case, let's walk through it. Okay, so we're talking about a cell, right? It has extracellular fluid outside, intracellular fluid inside. Okay. And what we know is that our electrochemical driving force for sodium points in. Okay. And so we're asking whether movement of sodium from the ECF to the ICF, whether that's active or passive. Right? We'll talk about it, but what we mean by that, right? Active versus passive. Active transfer is going to require energy input like ATP. Passive is not going to require an energy source. Okay? So in this case, right, this is going to be an example of a passive process because we've set up this gradient, right? This electrochemical driving force, this is our push, right? So this is describing our setup situation, this electrochemical driving force. If you think about it just in like easy terms, like normal everyday terms, the electrochemical driving force tells you which way sodium wants to go, right? So knowing that the electrochemical driving force pointed in tells you sodium wants to go in, right? Being pushed in, being pulled in, however you want to think about it, it wants to go in, right? So if we see a situation in which sodium is going in, that doesn't really require any explanation, right? Sodium wanted to go in, it is going in. We didn't need some additional energy to make it because that was the setup of this cell in the first place. We already had that driving force, that's where it wanted to go. So in this case, and we have just this much information, we're looking at a passive transport example, unless we have other information. Maybe you guys were moving forward thinking about different types. But basics, if something is going the direction it seems like it should go, the direction it seems like it wants to go, then that should be passive, unless there are other pieces to the puzzle. So for passive transport, we're describing something that happens spontaneously, right? So like automatically, you don't have to add in any cellular energy. So you don't have to add in any ATP. You don't have to harvest energy from redox reactions and high energy electrons like we do in mitochondria. None of that. It's just movement downhill along our chemical gradient, if we're thinking about something that's not charged, like glucose or oxygen, going to be typical examples of things that we'll be thinking about, but could happen with an ion as well. Okay? So we're going to see some types of passive transport. Right? So passive transport is a big bucket. Anything that doesn't require the cell to use energy to set this up. Okay? But in that bucket, we got whole, little smaller, I don't know, water balloons. So we're going to have simple diffusion. We're going to have facilitated diffusion, and we're going to have diffusion through channels. We'll take a look at each of those. So in simple diffusion, we're going to be thinking about things that go directly through our cell bilayer, right? So we remember that we have phospholipids with polar heads, nonpolar tails. We got a bilayer, so we got two layers facing each other, and it's like that all the way around. Okay? I'm not going to draw more, but it would be like that all the way around. So in simple diffusion, we're going to be thinking about something that can go through that lipid bilayer. So we're already thinking things that are nonpolar, potentially other lipids, maybe nonpolar gas. So like oxygen would be an example here, or another fatty acid might be an example here of something that's capable of crossing, right? So the membrane is going to be permeable to oxygen. We don't need any proteins because we can have that molecule, particle, whatever it is, going straight through, squeezing between our different phospholipids, right? So no membrane protein needed, goes straight through the bilayer. It's going to be going with its electrochemical driving force, but in this case, we're going to be thinking about nonpolar molecules because that's what can get through without a protein. So we're going to be thinking about a high-low gradient, a concentration gradient, because ions can't just squeeze through phospholipids, right? We only need to think about electrical driving forces and therefore electrochemical driving forces when we're talking about ions. If it's not an ion, chemical driving force is enough. Okay, so simple diffusion, we're just diffusing directly through the membrane. We do have some factors that can change how fast that happens, the rate at which that happens. Not every situation is going to be like 1 molecule per 1 second. So things that can change this are going to be the magnitude of the driving force, in this case, our chemical driving force. So what we're talking about is how big is this high-low gradient? Like how different are they? If we have a really high concentration outside and a really low one inside, it's going to happen a little faster. Right? So that would be a rate. Okay. And we also have membrane surface area. So when we have more membrane per volume inside, that is going to make this happen faster because this has to happen through passive process. So no energy. So we have to have molecules actually bump in to this membrane on the outside. So high surface area is really good for having that happen by kind of random chance. So membrane surface area, when it's bigger, is going to lead us to a higher rate, faster transport as well. So in the future, we'll talk about like the shape of red blood cells. They're like really flat and disc shaped. That's part of helping them maximize their surface area. So lots and lots of oxygen can get in that. Finally, membrane permeability. Something might be, the membrane might be permeable to it, like it technically can get through, but not everything's going to get through as easily, as quickly. So that might also affect the rate of simple diffusion. So here we can see a relationship between our concentration gradient, which our CDF is going to be the only driving force that we have to worry about when we're thinking about things that the membrane is permeable to, right? Because we already know that they've got to need to be nonpolar, otherwise that lipid bilayer is going to repel. Okay. So our concentration gradient is going to tell us the CDF, and that's the only thing we have to worry about with these specific molecules. Okay. So fundamentally, if our concentration gradient goes up, right, our net flux goes up. Very simple, right? So as the difference between inside and outside gets bigger, so does how much of that oxygen is going from outside to inside. More oxygen outside, less oxygen inside. The bigger that difference, the faster you're going to soak up oxygen. Here we see an example of like surface area and how they can affect the grade. So in this case, we're looking at, I think maybe some lung tissue, looks like to me. Should have labeled it, right? But you can see how this tissue has a lot of surface area, right? So like a lot of space where you can cross this membrane. So that's going to be easier to transport stuff than if we had less surface area, if we have less membrane, where we might have oxygen and the inside of a lung cell coming in contact. So when we get to the lungs, we'll see that they're structured as a bunch of like tiny little air bubbles with tiny little capillaries interspersed throughout, and that's why we're maximizing our surface area. So here we are looking at the membrane permeability and how that affects our net flux, right? So we're still thinking about something that can cross the membrane. So something that can get across our phospholipid bilayer. Okay? But if that molecule only has some low permeability, right? The membrane is only a little bit permeable to that molecule. You're going to see that the net flux of that molecule, so the speed basically at which it moves across the membrane, is going to be kind of lower, right? So both of these lines, as our concentration gradient goes up, so does the movement of molecules. But with low permeability, that process happens slower. If our membrane is highly permeable to that molecule, right, it goes up way faster, right? So in both cases, our concentration gradient as it increases changes flux, but permeability influences how the slope of that line is changing. So examples of what might affect this membrane permeability to a molecule are going to be the lipid solubility of the diffusing substance, which means like how nonpolar is it? How much is it repelled or not repelled by those specific phospholipids, right? So some things have some lipid solubility, but not that much. Some are very lipid soluble. There's a little bit of a difference possible between different molecules. We also have the size and shape of that diffusing particle. Basically, like big chunky things are going to have a little bit of a harder time crossing a membrane, even if they are lipid soluble and should be able to do it. In general terms, if we were thinking about chemistry of the system, we also include the temperature, because temperature basically means more molecules are vibrating and moving. So temperature would increase our rate of transport here. However, in your body, temperature is a regulated variable. So that's one of the things that we're maintaining through these homeostatic mechanisms. Remember, homeostasis is trying to keep us near a set point in a little regulated range. So your temperature should not be changing enough that it has an effect on diffusion. Right? That would be really bad. So hopefully your body's doing its job. You're staying in this homeostatically regulated temperature range. Finally, the thickness of the membrane. So we usually just think of our simple, basic phospholipid bilayer, those two little lines of phospholipid. But in some cases, we're going to have like mucus or water or other things kind of adhering to a membrane, functionally making it a little thicker, right? So we can imagine, right, a situation where not only do we have to cross our phospholipids to get in, right? We also have to cross some other layer kind of sitting on top surrounding the cell, right? Like stuck on. So in that case, and we got a bigger membrane, basically I have to travel farther, so that would make this process slower. Here is an example related to oxygen, right? So oxygen is a molecule that should be transported through simple diffusion. So in this case, we're thinking about how it travels in the lungs to the blood. Okay? We're thinking about a situation where we have water collecting in the lungs. So your lung, maybe you're kind of like drowning. You have fluid accumulating. What's that doing to the oxygen transport? In this situation, when this happens, oxygen isn't getting into your blood as effectively. We want to think, why? What's the problem here? Okay, yeah, that's why I was drawing the line in blue and being a little cagey on the previous slide. So one of the things that can act as essentially having a thicker membrane is a collection of water along the outside of the cell. So water can act like an increase in membrane thickness. There's kind of more distance, more thickness that we have to travel through. Again, more questions from your book to make sure that you're on board with that process. So a reminder that you have as you're reading some of those checkpoint questions. So with simple diffusion, we were thinking about molecules that can go directly between the phospholipids. We don't need any protein present, right? So simple diffusion is one type of passive transport. Our second type of passive transport is facilitated. So facilitated means like made easier, right? So facilitated discussion or make it possible, right? So in facilitated diffusion, we got the same setup, right? So we are still going to be going the direction that our molecule wants to go, right? That doesn't change. But what changes is that we do actually need a protein. So we need a protein there to make it happen, to make it possible, make it easier. Yeah. So in this case, we're thinking about things that aren't lipid soluble, and that the membrane is not permeable to, right? They can't just squeeze between those phospholipids. They're not small and non-polar. So we need a protein. Okay. So this is an example, one of the transmembrane proteins, right? So transmembrane goes all the way across, right? So in this case, we're looking at a glucose carrier protein. Okay? So you can see that transmembrane protein goes all the way through from the outside to the inside. And if we're thinking facilitated diffusion, we're thinking carrier proteins. So we're thinking about proteins that are literally going to carry our molecule across. Okay? So these carriers, to do that, are going to have binding sites. So here we can see that there's a binding site for glucose. It's kind of deeper in, right? So there's a place that glucose binds in on the outside by the extracellular fluid, right? So this blue part is the ECF, the extracellular fluid, okay? And when that glucose binds in, we're seeing that this carrier protein flips over. It changes shape as a conformational change. All those things mean the same thing essentially. All right, so when glucose binds in, this changes shape and glucose is flipped to the inside and we spit it across to the other side of the membrane. So that's how carrier protein works. That's what's happening in facilitated diffusion. So this is going to happen one at a time, right? So you're going to have one glucose binding in on that side where the extracellular fluid is. So glucose is coming in on the outside of the cell, binds to our carrier protein. Once we have one glucose in there, that carrier protein changes shape and spits glucose into the cell. So one side at a time. And that's important because that has implications for the rate at which we can see transport, right? So this is going to be still happening spontaneously, still happening passively, but we're not going to see the same relationship between concentration gradient and net flux as we did in simple diffusion, because we've got an extra complication here. We've got our actual carrier protein involved in the process as well. So if we're thinking about our net flux, so the rate of movement essentially across this membrane, we do still see the concentration gradient having an effect, right? It's not that this doesn't matter, right? So concentration gradient is still here on our x-axis. We're still looking at our net movement, our net flux on the y-axis. And we can see that the concentration gradient, as it goes up, so lots of glucose outside the cell, only little inside the cell, has an effect, right? So in general, when the concentration gradient is higher, our net flux is also higher, right? But we can see that this isn't a one-to-one relationship. It's not just like a straight line. And you can see there's kind of a cap on it too, right? So you can see how this line plateaus up at the top, right? So that's the difference that we see because of this involvement of the carrier protein. So when we think about the rate of transport in facilitated diffusion, we're going to need to think about that carrier. Right? One thing that's going to be true, the first carrier protein, so that glucose transporter, that glucose carrier that we saw, it's going to have a certain speed at which it works, right? Like it's going to take a certain amount of time to flip over, right? And take the glucose to the inside of the cell. So that's one piece of this puzzle. And that's going to essentially put a limit on part of it. Another thing that's going to matter is how many of those carriers we have, right? So if you imagine a cell, I'll imagine two cells actually. Two cells next to each other here. And if you imagine a cell that has just one glucose carrier versus a cell that has like four, right? If you have a high glucose concentration gradient, right? So in both cases, we have lots of glucose outside and only a little glucose inside, right? So we have enough concentration gradient that glucose wants to move. Okay. In the case where we only have one little carrier here in the membrane, you're only going to get one glucose through per however many seconds it takes for that glucose transporter to work. But in the same amount of time, if you have four carriers in the cell, you have four of them going across, right? So this is going to be much faster. This is going to be a higher net flux, a higher rate of transport when we have more of that carrier versus if we have fewer, right? So those are essentially how many doors are open for that molecule. So an example of this really exists in your body. So one molecule, or sorry, one way that we move glucose across cell membranes is through a transporter carrier protein called glucose transporter 4, so GLUT4. And GLUT4 expression, so like how many of these you have in a given cell on its membrane, isn't static, right? So it's not the same all the time. It's not the same in every cell, right? So the way it works is you can actually make GLUT4. So you synthesize it with the ribosomes and the rough endoplasmic reticulum. So you're taking a gene, you're reading it, you're making this GLUT4 molecule, which is a protein, right? So then it's going to get packaged into a little baggie. We call these cellular baggies vesicles. Okay? And in the presence of insulin, this is going to get inserted into the membrane. So we can imagine a situation, right? We've got our cell. We'll keep it with that first example, right? So like have one GLUT4 to begin with. Okay. But in the presence of insulin, right, we're going to have a lot of cellular machinery working. So I'm going to draw a nucleus. I'm going to draw some endoplasmic reticulum, right? We're making proteins in here because it's the rough kind. It's got ribosomes on it. So we're making a bunch of little glucose. They get packaged into a vesicle, right? So into a little bubble, okay? And then when we get a signal from insulin, which is a hormone coming from the pancreas, what the insulin tells the cell to do is to take that vesicle, bring it up to the membrane, right? And then this bubble pops, right? So we go from having this complete just circle here, this membrane. I'm gonna take an eraser briefly, redraw this. Okay. So we've got now the original membrane that we had, but that popped vesicle is inserted onto it, and we've got those GLUT4 transporters that were in the vesicle now on the surface of the membrane. All right. So this is how, in the presence of lots of glucose, we can speed up how fast your cells soak in, right? So insulin is released in order to soak out more glucose, like while you're eating food. Okay? So this is really helpful for glucose regulation. Glucose blood levels are one of the things that we should be regulating through homeostasis. Okay? And one of the types of diabetes that we have, right? This is part of what's going wrong. So in diabetes mellitus, when we don't have insulin acting on these cells, it means that this process isn't really happening when we get lots and lots of glucose surrounding that cell. So we don't start soaking up that extra glucose really fast, which means that you have a lot of glucose just kind of hanging out left over in the blood, which means your blood sugar goes up. Right? And that's a problem in various ways over time. So this is how we can change the rate of transport of a given molecule in different situations by changing these transporters or carriers and how many of them are present in these different situations. Even when this is still just pre-facilitated diffusion, this is still just a passive process. We're not using energy to pull these molecules in this example. Now, another type of diffusion we'll see that's really similar is instead of using a carrier protein that carries the molecule across, have a little pocket and flips, we can have diffusion through channel. The channel you want to think of as kind of like a pore, think of it an open door or an open window. This is still going to be made up of a transmembrane protein, but in this case, it's just open on both sides. All right, so here we see our channel proteins. Right, so they're in purple, they're transmembrane proteins, they go all the way across, but we can see that hole in the middle, right? So that's our open channel here. So this is going to act as a passageway for specific substances, right? So we remember that proteins have lots of different functions and lots of different shapes because we have pretty different amino acids that get folded up in all sorts of different crazy ways, resulting in different chemical and physical properties. So you can have a channel that lets sodium through. You can have a different channel that lets chloride get through. They're not like universally, absolutely everything can go through that specific channel. They're going to be specific to specific things. So some examples. We briefly mentioned aquaporins before. Again, we're mainly going to think about those when we think about kidneys. And at baseline, we're just going to assume any cell I tell you about has an aquaporin in it. That's a pore for water. So that would be an example of a channel. Substance specific, the only thing that moves through it is water. And like all the cells in your body, basically. We'll also have a number of different types of ion channels, and we'll be thinking about specifically where they are and why and what they're doing as we think about the structures of neurons. But we're going to have some channels that are like kind of always open, letting stuff leak across. We call those leak channels. We're going to have gated channels. So those are channels that can close and open under certain conditions. We'll talk a lot about voltage gated channels in the future that are triggered by different voltages, so different charges. Those are a big example of when we'll see these in action. We'll also have some channels that are bidirectional, not all of them. Bidirectional meaning we can go both. So different channels act differently, function under different conditions, but all of them, when they're open and working, let stuff go through. So when we think about how this affects our clock and how this affects our rate of transport, right, we're still going to be thinking about concentration gradients, all that good stuff that we did in the other ones. Now, like with the carriers, we're going to be thinking about our transport rate of each of those channels. So you can think of it as almost like how narrow or wide that passage is. It's not exactly that, but has a transport rate associated with that protein. Number of channels in the membrane is gonna matter too. So just like we could put more GLUT4 carriers in a membrane to allow more glucose to travel, you can put more sodium channels in a membrane to allow more sodium to travel faster. And finally, when we think about our ion channels, we are really going to want to think about whether they're open or closed. In particular, we're really thinking about gated channels when we say that for the most part. If they're closed, they can be present, but that's not going to do anybody any good. They have to be open to actually have an effect on transport, to have an effect on the movement of our ions. Good. I think we'll end there because that's kind of the end of our passive transport. I'll do a couple more examples on Wednesday and kick off with active transport.